Hello everyone, my name is Thanh Binh Nguyen from the University of Queensland, Australia. Today, I would like to share with you our recent publication with the title LEGO-CSM, a tool for functional characterization of proteins. This paper is published in Bioinformatics June this year. The paper is share first authorship between me, Alex, and Carlos. So let's go to the part that why we need this tool. Many of you might heard about the next generation sequencing, which is a technique that can zone the DNA RNA sequence. Thanks to the technology, the human genome cost gradually dropped from 100 million in 2001 to just less than 1,000 in 2020. And because of that, we know a lot of mutation in various diseases and its help for the treatment. As a consequence, in UniProt, a database for protein sequence, there is 230 million of protein sequence available. However, only less than 1 million of it is manually annotated and reviewed. Why? Because the experimental methods for characterizing protein function is time-consuming and expensive. There is computational methods that can use to predict the protein function. However, most of that is sequence-based only. Although the performance is good, but it did not consider the advance of structure-based. Most of you may know that the protein only function when it's full. So without consider the structure information, it might lose some of the important information. There is the structure-based method. However, most of that is based on high-quality structure. For example, X-ray. In 2020, AlphaFold announced as a solution to a 50 years old grand challenge in biology. In 2021, AlphaFold 2 were released and for published use. That's why we come up with our methods using AlphaFold, try to predict subcellular localization, enzyme commission number, gene ontology, or in short, LEGO-CSM, where CSM stands for cutoff scanning magic, which is the cutoff to calculate the distant pattern of atom in the protein structure. So first, when we have the data, we will check for the sequence in the UniProt as well as the structure from the AlphaFold. After that, we do the feature engineering. If that is the sequence, we consider the amino acid composition, the disorder propensity score, as well as the physical chemical property. If that is the structure, we will look at the graph-based signature, which consider the distant pattern of atom in a protein structure, or the residue depth, or solvent accessible surface area, or the secondary structure distribution and torsion angles. And after that, we apply machine learning method onto the feature that we have. As most of the people who are interested in the unknown or uncharacterized proteins is biologists. So we build a web server so that it's easier for them to use. So now let's look at the prediction for subcellular localization. We consider 10 different locations was listed here. This is the data set for the training where we have 11,000 data points, and in the blind test where we have 2,700 data points. And as can be seen, the data set is in balance, where most of the data is in nucleus. Very little data in peroxisome. It is very similar into the blind test set. As we have 10 different locations, we use multi-classification for this prediction. This is the performance of the state-of-the-art method, and deep block outperform on of this method. Comparing to LEGO-CSM, we better in the training set, but we worse than in the blind test set. The accuracies, both in the accuracy and in the Gorotkin, where it is the Matthew correlation coefficient in the multi-class classification. Now, let's look at the enzyme commission number, where it have 4 numbers to predict. The first number is the make class, and this is the list of the make class, where we have 7 make class, but we only predict on 6 make class due to the limited data. And this is the data set that we have. This is the sequence base. For the training set, we use the UniProt data, which remove 90% sequence redundant. And in the blind data set, we use the ECCB data set. Those data set in the blind data set would have 50% redundant to the blind data set were removed from the training set as well. Then we're looking at those sequence. If it have the available structure in the AlphaFold database, we use it for the model of sequence and structure base. And this is the number of the label that we have, including the non-enzyme term. We have 859 label need to predict. So the aim of this is, suppose you have a protein, then you need to ask whether this protein is enzyme or non-enzyme. This is the level 0 of the prediction. Then if it's an enzyme, it will predict the first number or the level 1. And it will go further up to level 4. The most difficult level to predict is the level 4, where all the 4 numbers need to be correct. And hence, as is the hierarchical, so we use the stacking for the prediction. In the performance, we compare the ECCB with all available method, both in the sequence and structure base, and ECCB outperform all of them. Comparing to LEGO-CSM, we worse than ECCB from level 0 to level 3. However, at the most difficult level, we better than them in terms of the Matthew correlation coefficient. Similarly to the sequence and structure-based model, we outperform ECCB. And as you can remember that the data in the sequence and structure base is just 1/3 of the sequence base, but the performance is very similar, which means the robustness of incorporate the structure information into the prediction. Now let's look at the last one, gene ontology, where we consider the cellular component or CC. Majority is cell part, organelle, followed by macromolecular complex. Biological process or PP, where majority is cellular process, metabolic process, biological regulation, and so on. And molecular function, MF, where majority is binding and catalytic activity. So this is the data test for the GO. of the sequence base, and you can see that BP, CC, and MF have a very similar number of data. The sequence and structure base have slightly smaller than the sequence base only. And this is the number of label for each classes. And as the BP is the number of BP, CC, and MF very similar, but you can see that the number of label in BP is 5 times more than CC and 4 times more than MF, which means BP is the most difficult case to predict. Now let's look at the performance. Before that, we need to look at, we use the binary classification for the prediction. So let's look at the performance. This is the benchmark of those state-of-the-art method, and DeepGo Plus outperform all of them. Comparing to LEGO-CSM in the sequence base, is outperform DeepGo Plus. The same for sequence and structure base is outperform DeepGo Plus. So as our methods is robustness, we want it to publicly available for users. So we build a web server. This is the link of that. To submit a job, you need to have a protein sequence. Is it compulsory? If you want to upload the PDB structure, you can upload it. Otherwise, we can search it in the AlphaFold database. You can put your email so that when the job finishes, it can send you a notification. Otherwise, you can refresh the page and then press submit. And this is the result page where it shows the prediction in the EC commission number, the gene ontology, the subcellular localization, and it shows some of the protein sequence feature as well. So you may wonder how long it runs. So this is the summary of the run. If the protein is less than 1,600 amino acid residue, it will run around 20 minutes. If it's longer than that, it may run from 30 to 40 minutes. So in conclusion, LEGO-CSM provides a unified resource to integrate 3 protein function components using both protein structure and protein sequence and structure information. LEGO-CSM perform as well as or better than other methods, and LEGO-CSM is freely available web server. So I would want to thank the author of this paper, especially Alex and Carlos, who equally contribute, and my 2 boss, Bryce and Asha. I would want to thank other members in the lab. I would want to thank CASS to give me the fund to be here, as well as the confidence to give me the opportunity to present my work. So this is my Twitter name and my email address. If you have any question, you can ask me here, go to the poster section, or send me the email. Thank you very much for your attention, and I'm happy to take any question.